geologist, and she obtained her PhD in environmental geochemistry at the Spanish National Research Council. Her primary research interest is environmental biogeochemistry, where she seeks to understand coupled chemical, biological, and physical processes that govern the chemistry and the fate of chemicals. She integrates hydrogeological, hydrochemical, microbial, and mineralogical informations to identify and quantify the different processes involved in the transfer of chemicals within the different compartments of a natural system. Currently, she is a Maitre assistant at the University of Lausanne. And with that, we are very happy to welcome you to give us an overview about arsenic and its cycling in peatlands. So, all to you.
Okay, thank you very much. And thank you very much for inviting me to this seminar. Today I'm gonna talk about arsenic in the Mekong Delta in Vietnam. But just for introduce you in the world of arsenic, arsenic is the 20th most abundant element in the Earth crust. The most common forms of arsenic in the environment is arsenic-3 and arsenic-5, the oxygen ions, and also arsenic sulfides. So the most oxidized form is arsenate, and the most reduced is arsenic in sulfide. So where can we find arsenic in the environment? Almost everywhere. Arsenic is ubiquitous in soils, sediments, and water environments, and surface waters and groundwaters. So in natural waters, we can find mostly arsenic in their inorganic forms as arsenite and arsenate, and in general, under oxidizing conditions, we find it in the form of arsenic-5, whereas in reducing conditions, usually is in its reduced form, arsenite. Why is it important to study arsenic? Because it has been defined as the king of poison. Arsenic is a highly toxic element. It has been ranking #1 in 2001 priority list of hazardous substances and disease registry defined by the World Health Organization. The arsenic intake is via drinking water commonly from groundwaters. That is a major source of arsenic exposure to humans in different regions of the world. So then it has been stated that human exposure to high concentrations of arsenic present in drinking water was responsible for the mass poisoning of population in the human history. So then this poisoning could occur due to the consumption of large quantities of arsenic with low exposure time, or chronic poisoning from ingestions of arsenic contaminated water with long exposure time. In any case, exposure to arsenic is a hazard to health, causing a variety of diseases from different types of cancer or this Blackfoot disease or arsenic keratose. As it is highly toxic, the World Health Organization has established a limit for drinking water of 10 micrograms per liter. So arsenic in the environment could be present due to natural and anthropogenic sources. So then, for example, the weathering of rocks and minerals containing arsenic, geothermal fluids, forest fires, as well as volcanic emissions are important natural sources of arsenic in the environment. Anthropogenic sources are, for example, burning of fossil fuels, mining activities, or the use of arsenic, herbicides, and pesticides. So these are the main sources of arsenic in the environment and for the contamination of groundwaters. Among these different arsenic sources, the major source of arsenic on all continents is sedimentary formations, and this is followed by mining activities. But how arsenic is mobilized in the environment? So the mobilization happens when the arsenic-bearing rocks, sediments, and minerals interact with water, and then due to different chemical reactions, sometimes catalyzed by bacteria, the arsenic is released to the water and can be transported to groundwaters and surface waters. The contamination of arsenic in groundwater has been reported worldwide, and here you can see a map of the world, and you can observe that it's almost everywhere. And arsenic contamination of groundwater is estimated to be affecting 500 million of people around the world. There are 4 major geochemical processes that initiate the natural release of arsenic from rocks and minerals into the groundwater, and each of them correspond to a typical geological environment. So for example, we have the process of reductive dissolution of iron hydroxides that contain arsenic adsorbed onto them. And then is a typical process occurring in Holocene sediments in floodplain areas. Another important process in alluvium and bedrock aquifers is the alkali desorption, when iron oxides that contain arsenic are in conditions of high pH, desorption processes occur, and then the arsenic that was already absorbed is released to the solution and could reach the aquifers. Another context is in areas of geothermal activity due to the geothermal fluids that contain arsenic. And also another environment in which we can find arsenic is related to sulfide mining and mineralized areas. In these areas, sulfide oxidation occurs, releasing the arsenic of the sulfide. But let's see some examples around the world of these geological environments with high arsenic concentrations. So an example of hot spring waters with high concentrations of arsenic are the hot springs in Yellowstone National Park, in which the concentration of arsenic could reach concentrations of up to 10 milligrams per liter. Arsenic is a ubiquitous component of active geothermal systems where the deep geothermal reservoirs can contain mainly arsenic-bearing pyrite and arsenopyrite and other arsenides. Then the uprising of these geothermal fluids may mix with other cold shallow waters and resources, making them unsuitable for water supply. Another example, in this case related to mining activity, is the sulfide oxidation process, and is the case of the Iberian Piritic Belt in the southwest of Spain. This area has been exploded over centuries, and the arsenic in the waters is caused by sulfide oxidation or sulfide dissolution. The most common sulfide is pyrite in this area, but this pyrite contains arsenic. So then the pyrite is unstable in the presence of water and oxygen, and then it dissolves, releasing also iron 2 sulfate and acidity protons, but also the arsenic of the structure of the mineral, increasing the concentrations in surface waters and could reach also the aquifers. So another example is this alkali desorption process, and this generally occurs in arid regions, like for example, in the Pampean Plain in Argentina. Here the arsenic release happens in oxic and high pH waters that promote the desorption of arsenic from mineral oxides, usually iron oxide. And finally, the last geological environment that I mentioned before is the Holocene sediments of floodplains and deltas like the Mekong Delta, in which the reductive dissolution process caused the release of arsenic. And I will talk a bit more in a few slides. But today I would like also to show you how other sources and mechanisms could also release arsenic to the water in this type of environments. So then I'm going to start with the first part of this talk, and I'm going to explain to you how microbially mediated release of arsenic we study from the Mekong Delta sediments. So I mentioned before that arsenic was an important problem of contamination of groundwater around the world, but specifically in the Southwest and Southeast Asia is a big issue affecting millions of people. So the arsenic contamination in these areas comes from the weathering of the rocks from the Himalayas, and once it's mobilized, then it's transported by the major rivers in the region and then deposited and accumulated in deltaic areas and floodplains. In these areas, it's generally accepted that the burial of the sediments and the establishment of anoxic conditions drive to microbial reductive dissolution of iron oxyhydroxides and the release of the arsenic from the sediments to the porewater and groundwaters, causing this important problem of contamination in the aquifers. So basically, the model states that the influx of organic carbon caused the oxidation of this organic carbon coupled to the reduction of iron oxides. These iron oxides contains arsenic 5 adsorbed onto their surfaces, and then during the dissolution, the arsenic is released, and usually as the context is very reducing, the arsenic 5 is also reduced to arsenic 3. But we were wondering if could be other sources and mechanisms that release arsenic to the porewater and aquifers in these deltaic areas. So then to answer these questions, we went to Vietnam, to the upper part of the Mekong Delta, here close to the border with Cambodia, to an area in which it has been reported high concentrations of arsenic in the groundwater. So the concentrations are in general higher than 500 ppb, 500 micrograms per liter. And as I told you before, the limit established by the World Health Organization is 10 ppb or 10 micrograms per liter for drinking water. So then we selected an area close to the Basak River that is one of the main branches of the Mekong Delta in Vietnam, and we obtained sediments. We obtained some cores, and we preserve them in an anaerobic glad box under anoxic conditions. So, as I mentioned before, to study the mobilization of arsenic is caused by the interaction of sediments that contain arsenic and the water. So then in our studies, we need to characterize the solids and also the solutions to understand or try to find out which are the mechanisms that cause this arsenic release to the water. So then we characterize the cores, we characterize the sediments with different techniques like X-ray fluorescence to obtain the chemical composition. We also analyze the concentration of organic carbon by a TOC analyzer. We also did some synchrotron measurements of X-ray absorption spectroscopy to obtain the arsenic and iron speciation in the sediment profile. And the arsenic and iron speciation means that with this technique, we are able to identify in which mineral or solid phase we find arsenic and iron. For example, we are able to see that arsenic is associated to pyrite or is associated to organic matter, and the same for iron. We can distinguish if iron is in iron oxide or is a siderite or associated to organic matter. And we also did some sediment dating with carbon-14 for those layers rich in carbon and optical stimulated luminescence for silicate-rich layers. So this is the core or the plot of the core. So the core we obtained consists of 42 meters of clays over a sandy aquifer. And by the chemical compositions, what we observed is that sulfur and organic-rich matter layers hold the highest concentrations of arsenic. From the X-ray absorption spectroscopy results, those that indicated the speciation, what we observed is that iron is associated to oxides and clays, and also to pyrite and organic matter. But we can see like an evolution from more oxidized environments to more reduced as we go down in the iron phases. And the same we observe in the case of arsenic. So we find arsenic as arsenite and arsenate, but as we go down, we can find it also associated to pyrite and organic matter. And why this core, let's say, is like this. So to understand this speciation, or why this speciation is how we find it, is because it's the result of the depositional environment at the Mekong Delta. So in which there was a marine transgression that reached a maximum at 5,500 years before present, and after there was a regression. So if we check again the speciation of arsenic in the profile, and we associate it with the depositional environment, we can see that before the transgression, what we have is just the flood water from the Mekong Delta that brings arsenic-bearing iron-3 oxyhydroxides with the arsenic adsorbed. And then these sediments are buried during the sediment deposition. But later there is the marine transgression, and then that yields a paleomangrove layer in which arsenic is trapped in the form of pyrite and natural organic matter associated to arsenic. And that makes sense because at that point the mangroves were developed in the area where we took our sample, and then the development of these mangroves produced an input of organic matter in our sediments, and on the other hand, as the sea level was there, there is also an input of sulfide that could favor the sulfate reduction in the sediments and produce or form this pyrite that trap arsenic in the structure. So later, we have the marine transgression continues, and we have just shallow marine sediments in which arsenic is again associated to pyrite and also organic matter, but not so much comparing to the moment of the mangrove development. And finally, we have the current moment in which we have just a similar situation at the initial. We have alluvial sediments, and then the Mekong River just brings some iron oxides with the arsenic adsorbed onto the sediments. But let's go back to, now that we know the context, let's go back to the main goal of the study, that was to try to identify if there are other sources and mechanisms that could release arsenic to the pore water and reach the aquifers. So I already showed you that this is the profile of arsenic and organic carbon in our course. And as you can observe, there is the peat layer that is very rich in organic carbon, of course, and it contains the highest concentration of arsenic in the profile. So that indicates that during the moment of formation, this peat layer acted as a sink of arsenic. But then we were wondering that, considering that it's really rich in arsenic, if this peat layer could become a source of arsenic to the pore waters. Then we analyzed the arsenic in the different layers, arsenic in the pore water. And as you could see, again, the highest arsenic concentration in the pore water correspond to this peat layer at 16 meters depth, and also the layer right below. So if we consider that arsenic could be released or leached from this peat layer, maybe this high concentration that we find in the clay right below could be just explained by the migration of arsenic downward. So then the main questions we were wondering is does this peat layer release arsenic? And if that is the case, which is the mechanism of release of this arsenic from the peat to the pore water? And that were the objectives of our work. So then, to prove the potential mobility of arsenic from the sediment under anoxic conditions, we performed flow-through leaching experiment in which the different sediment layers were exposed to a continuous flow of artificial pore water. That is, we prepare a water in the lab with the similar composition of the pore water of the peat layer, or artificial groundwater that it was again prepared in the lab, but with the composition of the aquifer. So basically the flow-through experiments, you prepare your solution, and then there is, by means of peristaltic pump, the solution enters into your reactor, reacts over a period of time with your sediment, and then you collect the water result of the reaction. Measuring, for example, the arsenic in your output solution, you can be able to calculate how much arsenic has been leached from your solid. So these experiments basically provide information about the potential release of arsenic into the pore water. And you can observe the results on the right. And there are large differences in the total amount of arsenic release over 600 meters, depending on the sediment type or depth that we used. And in any case, the peat layer is the one that leached most. But then we also normalize the amount leached with respect to the surface area for each layer and with respect to the arsenic in the sediment. And as you could see, the peat layer releases more than arsenic than any other sediment layer, which indicates that could be a source of arsenic under anoxic conditions. So then once we knew that could be a source of arsenic, this peat layer, then we decided to try to find out if this release of arsenic was a microbially mediated process or just an abiotic process. So to check that, we repeat the experiments with artificial pore water and with artificial groundwater under the exactly same conditions, but in this case, we did the experiments treating the sediments with a poison with sodium azide in order to try to decrease or inhibit the microbial activity. So as you can see here, here are the results of the untreated sediments experiments. And if we compare with those poison, there is an important difference in the amount leached, which indicates that the process of release is mediated by bacteria or microbially mediated. So I know that I talked before about the arsenic speciation, but I would like to highlight which species are in the peat. So we find arsenic in this peat layer associated to pyrite and also associated to organic matter. In the case of pyrite, we found arsenic in euhedral pyrite and also in rhomboidal pyrite that were in the sediments. Then to investigate the potential change in the solid phase arsenic speciation during the flow-through experiments, we compare the initial sediment with the analysis with the same sediment after the leaching. Here I just wanted to show you that there is an important amount of arsenic leached during the experiments, but to really compare and be able to see if one of those species leached more than the other, we compare it in percentage. As you can see below in the lower part of the screen, the fractions of the two arsenic species after the flow-through experiments were very similar to those in the original peat. That suggests that comparable amounts of arsenic were released from organic matter and from arsenic pyrite. So far we knew that the peat could be a source of arsenic, and from the peat, both species could release arsenic to the pore water, or both released arsenic to the pore water during the experiments. So we have arsenic, the pyrite, and we have the organic matter, both sources of arsenic in our experiments. So then the next step was to investigate the composition of the pore water to try to hypothesize which mechanism could explain this release from these two species, from pyrite and from the organic matter of the peat layer. And then we saw that there is nitrate present in the pore water composition, although in low concentrations. So then we thought that nitrate could act as electron acceptor, and then we hypothesized that nitrate reduction coupled to natural organic matter and arsenic pyrite oxidation could be the mechanism that caused this arsenic release to the water. To test our hypothesis, we carried out batch experiments in which the peat sediment was amended with increasing concentrations of nitrate. And again, we did some incubations poisoned with acid to try to determine the role of the microbes in our experiments. So we also analyzed the sulfur speciation before and after the reaction, and this is just from the peat before the reaction, and we can see that sulfur is found as organic sulfur and in pyrite. We can also observe there is a small quantity of sulfide. And then we compare these results after 40 days of incubation, after our batch experiments with the different concentrations of nitrate. And you can see that there is a decrease in the relative contribution of pyrite to overall sulfur. So this difference is the amount of pyrite that had been oxidized during the experiments. So then we also studied the water composition during the experiments, and here I show you the concentration of arsenic with respect to the initial concentration of nitrate. So these samples were taken after 40 days at the end of the experiments. And as you could observe, there is a linear increase in the arsenic release from the sediments with increased the nitrate amended, which indicates that nitrate has a role in this release of arsenic from the peat. Also, we observe there is a decrease in dissolved organic carbon concentration with increasing the initial nitrate concentration. At the end of the experiment, nitrate was below the detection limit, so suggests that it was everything denitrified, so the nitrification occurred during the experiments. So combining all the information that, combining the information of the sulfur speciation and these results from the water, that confirm our hypothesis of the nitrification coupled to pyrite and organic matter oxidation, and then explains the arsenic release from the peat layer. And finally, the last step, it was to try to see if it was microbially mediated process or not. And the comparison of the experiments performed with the untreated sediments and those poisoned with acid indicates that this decrease that we observe in the poison ones indicate that the process was a microbially mediated process. And similar results we can obtain with the dissolved organic carbon. This increase suggests microbial degradation of dissolved organic carbon. So if we go back to the initial questions, does the peat layer release arsenic? We can answer to this question, that is yes. And we can also propose a mechanism of release of arsenic, which is microbial oxidation of pyrite and organic matter coupled to nitrite reduction. So now that we know that the arsenic is released from the sediments, that could be due to this iron reduction, but also could be other sources like these rich organic matter layers. The second part of this talk is about which processes could control the aquifer chemistry. Again, in the same area, we collected samples of groundwater and sediments from the aquifer. The groundwater samples, we obtained it from the wells of farmers, and we also obtained again the sediments drilling from the aquifer. We did different sampling campaigns over the time. In this case, the core consists of 7 meters of clays over a sandy aquifer, and we just characterized the aquifer because we wanted to study which processes occur in the aquifer. And then the mineralogy of this aquifer was mainly composed of quartz, mica, feldspar, and chloride, contains arsenic concentrations of 5.5 to 6.9 ppm, iron between 4.1 and 4.5%, and total organic carbon of 0.05%, which is relatively low, but similar to other aquifers of the area. Then again, we characterize the solids with X-ray absorption spectroscopy, and the speciation of arsenic results so that we can find it in the form of arsenic-3 and arsenic-5. The iron speciation indicates that we can find iron in the form of iron oxides and clays, and the sulfur speciation results so that we can find it in the form of elemental sulfur and sulfate. So then we monitor 14 wells over a period of time of 1.5 years, and we observed some seasonal variations in the groundwater chemistry. So we saw that there is an increase of the EH values in the sampling of November 2015. So the conditions were relatively oxidizing, and also an increase of dissolved oxygen in November 2015, which was consistent with the EH values. Arsenic values were lower in this sampling campaign of November 15th compared to the other two field campaigns. And regarding arsenic, regarding iron, there are some varieties. So some wells saw some patterns similar to arsenic, other no significant changes, and other an increase in iron concentration in November. These are those that has similar pattern to arsenic. Sulfide was only detected in some wells in May 2014 and January 2015. And finally, there is an increase of nitrate in November 2015. But in summary, what I would like to show you with these results is just that when there is no oxygen or the oxygen is very low, what we observe in general is that increase of the arsenic and iron concentrations, a decrease in nitrate, and a decrease in the EH. However, when oxygen is present, even at low concentrations, we can observe a decrease in the arsenic and iron concentrations, an increase of nitrate, and an increase in the EH. As previously reported in other groundwaters and in deltaic areas, during periods of groundwater recharge, oxygen and other oxidized chemical species could reach the aquifer, and then the concentrations of reduced species decrease. When the surface is limited, like here, then reducing conditions are established, leading to an increase in the concentrations of aqueous arsenic, iron, and sulfide. So then we hypothesized which processes occur to explain these seasonal variations. And then in the moments of that there is an input of oxygen in the aquifer, there is no sulfide, that we interpret it like a sulfide is being oxidized to elemental sulfur or sulfate. Iron 2 is oxidized to iron 3 and then precipitate in the form of iron oxides, and then arsenic could be adsorbed onto these new iron phases. Ammonia could be oxidized to other forms of nitrogen, nitrite or nitrate, explaining maybe this high concentration of nitrate. However, when oxygen is not present in the aquifer, sulfate reduction could occur, increasing the concentration of sulfide, so then we could detect it in the water. Iron oxides or hydroxides were reduced, releasing part of the iron and also the arsenic. And nitrate could be reduced to nitrate or ammonia or nitrogen. So then we wanted to know if these processes explain these seasonal variations in the water chemistry, and which is the role of the microbial processes in all these seasonal changes. So to answer that questions, we did some laboratory experiments. So then with these experiments, what we wanted is to try to reproduce these biogeochemical processes and try to explain the variations in the chemistry of the groundwater, and also to find out if they were microbially catalyzed or not. And then we did what we call redox evolution experiments and amended experiments. And I would like to say here that part of the results I will show you from now to the end of this talk are part of the master thesis of Sophie Britange. Here is Sophie. She did her master thesis at EPFL in our group, and she came to the field to help us, and after she did experiments in the lab. So the first type of experiment, these redox experiments, were designed to study the evolution of arsenic and iron, mimicking the aquifer conditions where temporary oxygen is advected into the aquifer. So with that aim, a set of 4 experiments, a triplicate and 1 control poisoned were performed. And for that, we used some aquifer sediments poisoned with sodium azide and performed with autoclave artificial groundwater, similar to the composition of the aquifer, and 3 experiments with untreated aquifer sediments and unfiltered groundwater from the aquifer. So as you can see here, the incubation under anoxic conditions cause an increase of iron 2 over time. And if we compare the poison and non-poison, we can see that the poison experiment released a bit lower concentration of iron that could indicate that the sodium azide could have reduced the microbial activity. So this we interpreted as iron oxide reduction that we already observed from our SAMES results and X-ray absorption spectroscopy results that iron oxides were present. And then we also studied the arsenic concentration under these anoxic conditions, and we observed that has a similar pattern. It's released during this anoxic condition, probably because this arsenic was already absorbed onto the sediments, and then during this iron oxide reduction or dissolution, it's released to the water. So then we added some oxygen to the system, and we can observe that the iron decreased, and we interpret that iron had been oxidized to iron 3 and then precipitate, sorbing part of the arsenic of the water. After a certain period of time, once oxygen is consumed and anoxic conditions are established, an iron reduction starts again. So the experimental results suggest that the variation of iron and arsenic concentration in the aquifer over time responds to changes in the redox conditions between anoxia, which favor the microbial reduction of iron 3 hydroxides, and the arsenic release, and oxygen inputs with the precipitation of iron phases and arsenic sorption onto them. But what about the sulfur and nitrogen cycles? So to answer that, we did amendment experiments. For these experiments, we used unfiltered groundwater and non-poisoned sediments, and also we poisoned some of them, and we used autoclaved artificial groundwater. We used different amendments like sulfate, nitrate, sulfide, ammonium, and oxygen. So here you can observe the results of the sulfate amendment experiments. The addition of sulfate in the untreated experiment resulted in a higher decrease of the sulfate concentration when we compare to those previously poisoned, which indicates that microbially mediated sulfur reduction occurs during the experiment. So also to investigate if oxidative sulfur processes could occur, sulfide was amended to sediment and groundwater, untreated and already poisoned. And as you can see, sulfide decreased in all the experiments, also in that poisoned previously. However, in those not poisoned, this decrease of sulfate caused an increase of the sulfate in the solution, which suggests that sulfide is being oxidized in the experiments. And however, in the case of the poison 1, we observed that the sulfate decreased. And probably this is due by microbial sulfur reduction, indicated that the acid poison didn't work in the case of the sulfur reduction. In any case, the two different behaviors between the untreated experiments and the acid poison experiment suggest that microbially mediated sulfide oxidation occurs in the experiments. Here you can observe the results of the experiments amended with nitrate. And as you can see, anaerobic reduction of nitrate occur in the amended incubations, both untreated or poisoned. So from these experiments, we can only say that nitrate reduction occurred, but we cannot be able to distinguish if it was microbially catalyzed or abiotic process, for example, coupled to iron to oxidation. In this case, nitrite was detected in all experiments, but at very low concentrations. And finally, the last process we wanted to investigate is the ammonium oxidation. And the results show that in the ammonium amended experiments, the ammonium concentration decreased and decreased more if we compare to that poisoned, which suggests that the process is microbially mediated. But in this experiment, we observed something else because the amount of oxygen that we added cannot explain this decrease in the ammonia concentrations. So that could be that there is another process other than this aerobic ammonium oxidation consuming part of the ammonia during the experiment. Like, for example, microbially mediated ammonia oxidation coupled to iron 3 reduction. In this case, nitrate was not detected during the experiments, and nitrate was detected at low concentrations in the experiments performed untreated with the untreated sediments. So in summary, the results of our experiments could confirm that sulfur oxidation and sulfur reduction occurs in the experiments, and both are microbially mediated. And regarding the nitrogen cycle, nitrate reduction occurs, could be biotically or abiotically, and ammonium oxidation occurs catalyzed by bacteria. So in the presence of oxygen, we were able to confirm it, but it seems there is another process also anaerobic that consumes part of the ammonium. That could be ammonium oxidation, but we cannot be sure about this process. So in summary, the main findings of this work was the redox fluctuations in the aquifer appear to be related to seasonal variations in the hydrological system, and microbially mediated processes govern the arsenic concentration and the water chemistry in this area. Finally, I would like to highlight that this work is the result of the effort of many people and different groups, like my previous group at EPFL, people from CNRS, and also the help of the people from Vietnam. And finally, I would like also to acknowledge the funding from SNCF, from RECIF, and the technical assistance from the different synchrotrons, from SLAC and Electra synchrotron, and also the help of all the students and team from Vietnam that help us a lot during the field trips. Acknowledge also to my group at EPFL, the Environmental Microbially Group, and all the technicians from the Central Environmental Laboratory. And thanks to you for listening to me. Thank you.
Thank you, Marie, for a really nice talk. If anyone have a question, either you can raise your hands or type it in the chat. Either way, I can probably see you. Karim has a question.
Yeah, so so thank you, Mapi. I guess I would have many questions, but I keep some of them for later on for emails. First question is about this the organics in the peat sediments that you were mentioning, where you have a high concentration of arsenic. So you mentioned that they contain high amounts of thiol functions.
Mm-hmm.
And so just two questions. The first one is, I was wondering how, I mean, if that was a usual thing, or it was more like associated with the fact that they were deposited in the presence of mangrove to have such an amount of such a high amount of thiol functions. And the second question is, if I understand it well, what you say is that eventually the arsenic that is complexed or associated with these thiol functions is not released so much by it's not, I mean, what I could understand is that oxidation of pyrite was contributing relatively more than the degradation of these organics. Am I right? Or I misunderstood?
Okay. Regarding the first question, it's, if I understood your question, basically in this, this mangroves area that you have a lot of organic matter, the affinity of arsenic for the organic matter is really high. And the way to do the link is through this thiol bond way, I mean. But in this case, of course, we had a lot because it was very rich in organic matter. And that happened also in peatlands, in other areas. But on the other hand, arsenic has a lot of affinity for sulfur and for this organic matter. So it's what we see in the peat. We have associated to pyrite, and we have also associated to the organic matter. I don't know if I answered. Regarding to the thiol thing, maybe it was not clear from the X-ray absorption spectroscopy results. What we saw is that indicates that the amount is similar from both phases. So let's see if I find it here. So what we saw here is that before and after the reaction, the percentage is exactly the same. We were expecting something like, if it's pyrite, for example, pyrite decrease, or on the other way. However, the fact that we found exactly the same, more or less the same percentage before and after the reaction indicated that the concentration released for both species were kind of similar. And we did some calculations also. I don't know if I can, well, now I cannot show it. Can you see my screen now?
Yes.
Okay, because we did, I have some extra slides somewhere. I think it's this. So we were analyzing also with X-ray fluorescence, and we combined with this X-ray absorption spectroscopy results. We tried to calculate how much was leached from each species. And for example, associated to the organics was like 8 ppm, and the amount leached from the pyrite was around 6. So it was kind of similar.
Okay, and so so for the degradation of the thiol containing organic molecules, what kind of microbes do you need for that? Do you have any idea about what microbes might be more efficient, or might be particularly efficient, or maybe any microbe might do the job of degrading this kind of organic matter?
I don't know. I know that we tried to extract the DNA from the peat, and we had a lot of trouble. So at the end, we decided not to do it. We tried different ways and different times, and at the end, it was not possible. Maybe there are better techniques than we used, but we couldn't.
Okay. Well, while other people are thinking, I have a couple of questions, mainly on the side of the seasonal variations affecting the redox fluctuations, which you see in the aquifer.
Mm-hmm.
What's the groundwater pumping regime in this area? Like, Is it like, could the redox fluctuation could be tied to how the wells, like how much the groundwater is being pumped, and if it's extensive pumping, and that might induce some sort of fluctuations? Because
Yeah, it could affect, to
it's
the
right.
cycles. Yeah, I
Yeah.
agree. I agree. I don't know. I know that it is used by the farmers. All of them, they have wells. Actually, we took it from them. But hydrologically, I never did any check or any study. I didn't want to enter a lot about these cycles because we don't have the information. The only thing that we see is the changes in the oxygen content and other chemicals content. We don't know. Yeah, depends. Yeah, if they pump a lot, of course, it's gonna change all the hydrological.
Yeah, right.
I don't know. It's a bit tricky because in theory, the wells are really contaminated by arsenic. So even when you ask, they don't tell you because, for example, if they have rice, the rice absorb a lot of arsenic. So usually they don't tell you that they water with this groundwater.
Yeah. Right. I mean, that's like one of the big problems in Bangladesh, right? It's the whole the wells are fully contaminated, and over there, at least they know it's from the groundwater pumping regime. It induces a lot of fluctuations in how much arsenic is getting mobilized. So that's why I was just wondering if you saw something along those lines. The second question I had was about the denatrification processes you mentioned. Did you, in your lab experiments, did you see any sulfide, like any evidence like there was some sulfate reduction happening in your system?
In which experiments? In the
experiments, in the lab experiments where you incubated the peat sediments with the nitrate amendments?
The peat, okay.
I think it's yeah. So further up.
Here.
Further up.
Up.
Up. Yeah, these ones, these experiments. Slide 34.
34.
Yeah.
Sorry. No, we did 35.
34.
34 is this.
Yeah, so these experiments, if I understood correctly, you had the peat sediments and you put in different amendments of nitrate, and you were looking for evidence for denitrification. Is that right?
Yeah, the nitrification coupled to
I was just wondering if you also, because you mentioned that you don't know much about the microbial community habiting the peatlands, and I was wondering if sulfate reducers might be also present in your system, and if you saw some evidence of sulfate reduction, and when you measure the solution chemistry afterwards.
Sulfate reduction, you mean?
Sulfate reducers, yeah.
Yeah, could happen because we also measure the sulfate. I didn't show it today, but I have it somewhere. Well, so we measure the sulfate also during the experiments, and we compare. We did the same. And at the end, the mass balances of anything worked.
Okay.
So one possibility is part of the sulfate could be reduced by sulfate reducers, but I don't know much about the microbiology, microbial community in this field.
Yeah, no, I was just wondering, how does that affect the whole arsenic release if those mechanisms are happening at the same time, denitrification and sulfate reduction?
Well, it's could if some thioarsenate or other species could form, also will increase the mobility, but we never check that, so it was not possible. In the future, maybe it's a good idea to check this.
Thank you. Guillaume has a question.
Hello.
Hi, Guillaume.
Hello, Mappy.
Hello.
Yes, I have two questions. One coming on the one that Nea has just asked. One is, the first question would be on denitrification. So is it, I mean, we think it is coupled to the pyrite oxidation. So it means to sulfur oxidation, sulfide oxidation maybe, or iron oxidation. And what can we expect to do to distinguish between the heterotrophic and I mean the heterotrophic denitrification. I mean, starting from the organic matter that is present, or the denitrification that would be based on the iron, I mean the mineral oxidations, because these two processes seems to be running at together. Mm-hmm. And so it's difficult to imagine that we can have, I mean, how do you think it is possible to oxidize the pyrite? What would be the most efficient between the two? Can it go together? I mean, the oxidation of iron, for instance, and sulfur by the nitrate, mm-hmm. catalyzed by bacteria, together with the oxidation of organic matter, because there is also possibility of heterotrophic denitrification. So this is, can we couple the two? How does it? How can it?
Well, honestly, I don't know. I know that in these experiments, everything suggests that both are happening. I don't know if one is more favorable and occurs at the beginning and after the other. I don't know exactly. I don't know exactly how exactly goes.
Because there's one hypothesis for mineral denitrification. I mean, mineral assisted denitrification would be that the first step is the nitrate reduction to nitrite. Yeah. And then the nitrite is able to react with iron 2, for instance. That has been more or less demonstrated by the Kepler groups and mm-hmm. people like that. And also we worked on this kind of processes in IMPMC2. So there are different steps in the denitrification process. And maybe one of the steps, the first one is really difficult. And the nitrate reduction is really difficult to obtain only with minerals, but maybe pyrite can do it. I mean, that was kind of my question. And the second point is, after you have this nitrite produced by the bacteria, then you can have many inorganic reactions that can lead to the to production of nitrogen, for instance. So second question is the speciation of arsenic is. Okay, we have this distinction between thiol-bound arsenic and the pyrite, arsenian pyrite. So there are maybe some space for other species, like has been described in other peat, like the people found. Yeah, railgar, or maybe amorphous orpiment, or those kind of things. So, do you think it could be possible to see it in our system, in your system? I mean, do you think from exafs we are enough sensitive to really distinguish between thiol-bound arsenic-3 and amorphous orpiment, for instance, in this system?
We were not able to distinguish, but I guess if it's in low concentration, like less than 10%, probably if it's there, that could be there. We were not able to see them
because the spectra are very similar. I mean, so it's really it has been discussed in the paper by Langner. And they did this natural geoscience on that, and they said, okay, with the wavelet analysis, we can see there is difference between the amorphous orpiment and the arsenic bound to glutathione, and that's the same reasoning that we used for analyzing the data here. But my feeling is that it is, I mean, mostly it's this thiol-bound arsenic, but I think we can't really exclude, as you said, the presence of other phases, and that could be maybe more reactive than the pyrite, and could also. Anyway, I'm still surprised by the fact that we have the same reactivity for the thiol-bound arsenic and the pyrite in the system. This is really surprising that there is no specific way of, I mean, but okay, maybe this is the way it works. But anyway, it's difficult to imagine that the reactivity of the two systems will be exactly the same react with respect to nitrate. But it's not impossible. I mean, it's not. Okay. Thank you anyway for the very nice talk. Thanks.
Anyone else? Karim, you have a question following us?
Yeah, so a few more. Yeah, so in the second part, I mean, at some point you saw you showed the cycling between oxic and anoxic conditions, and so two questions about that. The first one is that so you see a reoxidation. I think it was really in the beginning. You see, you show the yeah, so yeah, that might be this 65 or something like that, right? Yeah, 65, for example. Yeah, so I was wondering, so have you tried to cycle it several times? I mean, I was wondering, so looking at the one on the right, I was wondering, so if the rates are just stepping down somehow, I was wondering if, you know, what would that mean, I guess? Do you build up some kind of reservoir that is less and less reactive or so on?
Yeah, I didn't try, but there are papers regarding several cycles of arsenic and how they react, and in general, in experiments in the lab. And they could see this decrease in the arsenic release and sometimes also in the iron. There is a paper of Parsons 2013, I don't remember. They say that could be for different reasons, changes due to the redox cycles, to the crystallinity of the iron oxides, the mineralogy of the iron oxides, the microbial activity could change also over time, or the labile organic matter, maybe it's at the beginning more labile, and after what it's the leftovers are less. But it's not new. This has been observed in other systems.
Okay, and so the second question, and that will be my last one, but so when you look at the iron, it looks like you reduce iron even in poisoned samples. I mean, I understand it's less than in non-poisoned samples, but there's still some reduction, right?
Yes. After doing a lot of experiments with these samples, we observed that we have to be careful with the poison we used. For example, the sodium aside, I didn't show it today, but the sulfide reduction, we observed perfect sulfide reduction after poisoning with a lot of acide sediments. So then we have to switch to mercury. Actually, I only saw the results from mercury because with mercury, we were able to see differences. Our guess is like this anoxic processes for acide is not the best poison. It's better to use a different poison like mercury, or at least for us in this system, we have to repeat some experiments with mercury because of that.
Okay, so you think that the poison is not killing the cells somehow?
No, I think they could decrease a bit the effect, but no, they don't kill, no. They actually, in many of the processes, we can see things that happening. So in this, in sulfur, we can say the sulfide oxidation, but the sulfide, you have, we don't know, but it seems that there is like a sulfide reduction and they were poisoned previously. So no.
Thank you.
Okay, with that, just checking if anyone has a last minute question. Okay, I guess yeah, Justin. Oh, does anyone? Elodie, are you? Oh, okay. For a second, I thought Elodie had a question. Okay, well, with that, I guess we're in time. So thank you so much, Mary, for a really good talk and a lot of information on arsenic. So really exciting. And thank you so much. And thank you all for attending, and see you